what is foreign. And microbiome is the first layer of barrier and first layer of protection that comes into picture and gives us the natural defenses that's present in our body. And this microbiome sort of trains our immunity to know what is good and what is bad. So it just doesn't start killing everything left, right, center. And this is why microbiome and immune system in together, in conjugation, is the way to understand immunity in humans and in any animal forms, to be honest. In the past, we have shown that this microbiome, this association of the microbiome with the immune system, it starts quite early in human life, especially during gestation, during second trimester of gestation. There are microbiome, like there's microbes present in our body, in mother, which gets transferred from the mother to the fetus in different organs of the fetus, and it sort of trains the fetal immunity in a way that it retains a memory for these microbes. And it's not just us, but several other studies in the past decade have actually shown the impact of microbes and microbial products, metabolites and antibodies which are in response to microbes. They help to shape the immune diversity in the fetus. And that's why it becomes important to study this, because that's the start of immunity. And that's where the immune system is getting educated to face what's going to come outside later when the baby is born. And this is in a way kind of natural immunization, which scientists have been believing since hundreds of years, that what happens during gestation and right after that is sort of providing natural immunization to the baby. Now, what we are doing currently in the world is basically trying to also understand the natural immunization, but also try to boost the immunization process, and that is what the vaccination comes into picture. So when a baby is born, there are myriad of vaccination, immunization schedules which is given to the baby in order to fight the microbes that might come later, even though they are not exposed at present. But in a way, if you really see, these two are not mutually distinct entities. They are quite intertwined. So we have certain level of natural immunization, natural immunity from the mother and from the environment, and then we combine it with the vaccination, and there we are, protected, nice and happy. But like Shyam mentioned earlier, disparity is something which holds us all together to study these kind of systems. And in this system particularly, where there is a baby involved who is this is the most vulnerable population in our world, there's a huge disparity across world in the response of these babies towards vaccination, especially if we have to divide the world between low income and high income settings, or the settings where there's a lot of migration going on, or different food habits. There's a huge disparity here, and we still don't understand how these vaccines work, but we are still giving the same kind of vaccination without any booster, without any adjuvant to all these babies, and we don't see the response in all the time. So this is a time to understand what's happening, what can we do to enhance this process in a way that the babies are more protected in areas where they are actually less responding. So the idea here is in this whole microbiome diversity atlas to study the microbiome of the newborn, and this is where I come into picture because I'm interested, my lab is interested in newborn immunity and microbiome. But in a way, it's open to all sorts of ages and all sorts of settings. But we can start with the microbiome in newborn, how it develops across time during gestation and then right after birth, and how it impacts the vaccination response in early life. So here, the kind of sample and the kind of assays that we'll be doing is we'll collect stool samples and we'll do metagenomics, we'll do bio, because by that we can sequence the entire 16S ribosomal RNA and get to know the diversity up to species level. And we will also collect some blood to study the antibody titer of the response to certain vaccines, and also the neutralization ability in our immune system. So the vaccine that we have chosen currently, and of course it is open to the more the merrier, but right now we are focusing on hepatitis B vaccination, which is given to the babies when they are just 12 hours old. So within 12 hours of their birth, this vaccine is given to the babies. And then the idea is to collect the blood samples after a few days and look at the antibody titer and neutralization assays, and also look at their microbiome over time. And then sort of look at the correlation between if there is a certain type of microbe associated in early life, they might have a better immune response in this case. And then we start with Hep B, and the idea is to expand to more early life vaccines. Now consider a different setting. So this is where a healthy baby gets the vaccination within 12 hours of life and is quite protected, depending upon which part of the world the baby is in. But what happens if the baby is born preterm? Irrespective of which part of the world the baby is in, if a baby is born preterm, they are always vulnerable to infections. Now it depends upon the kind of infrastructure and the kind of hospital facilities are there in those countries, but the baby inherently is quite vulnerable to infection. And these babies have to wait for even immunization. They don't get immunization right away. So they are kept in enclosed containments, and then they get immunization only after they are healthy. So they were vulnerable to start with, and they get further vulnerable because they cannot take vaccination at such early time. So we are taking baby steps in this direction as well, and with our collaborators, we are building this cohort. So with our collaborators in Australia and Singapore, so we are building this cohort where we are trying to study the preterm and term babies' microbiome, and eventually the idea is to bring in the blood aspect as well. So what happens when a baby is born preterm? A lot of times the babies are given probiotics, and studies have shown that giving these probiotics actually increases the risk of fighting towards infection in this baby. Not the risk, but basically it's protected. If the probiotics are given, the baby is protected and they don't get infected as easily as a preterm baby who didn't receive probiotic. But these are very early stages of studies and we don't understand how that happens. So we are collecting longitudinally the full term and preterm cohorts for 1 week, 4 week, 4 month and 1 year of life, and we are sort of understanding the comparison between these microbiome and the dynamic changes in these microbes. And the early data shows that, I know it's a busy slide, I don't have much time, so just focus on this one. So the first column represents the preterm babies that don't have probiotic, and the second column is the preterm babies that have been given probiotic. And these babies have certain developed eventually with time. By 4 months, they have developed certain microbiome, which is very good for fighting infection for the baby and also for metabolism, proper metabolism of the baby. And it basically represents something similar to what a full-term baby has. So we are kind of trying to skew the microbiome of the baby of a preterm baby towards a full-term baby. So it's more like sort of supplementing something so they can be better protected. Now the idea is, if the blood response also looks something similar, if probiotics are given, or if vaccines are given along with probiotic adjuvants, which we have seen in the cases of rabies vaccination and in certain other vaccinations, certain probiotics and metabolites like retinoic acid have been given, and they have proven to be really good. So this is the plan, and then if you really focus on these babies up till 1 year, there is actually no difference between the preterm and term baby, while there is a huge difference between the preterm babies that were not given probiotic. So I think my time is over. The idea is so right now we are starting from Asia Pacific, but the idea is to expand this and look at the global microbiome development and also across ages and across different ethnicities, different populations, and eventually just go global with this. And again, the idea still remains to understand what happens during development and how this development sort of entails the way the human being or the baby or the adult responds to infection and everything. So this is a collaborative network at present, and this could be you. And if you work on any disease, you work on any sort of developmental aspects or the disease aspects, please feel free to talk to us. We also have a microbiome breakout session after this, so please come and talk to me, and it will be great to have some discussions around this. Yeah. Thank you.